All right, well, I saw Stuart walked in, so I said, now we can actually start. Well, it's a first of all, to have everybody come to the room, having people in person. We are recording this, so people who want to watch it later can do it. Are we also live streaming it? No. So we're recording it so people who miss it can watch it later. But it's also a pleasure to introduce one of our own. So Josh Hihath, is that how you pronounce it right?
Close enough. Hihath, yeah.
Hihath, has been, he started here. He was an assistant research professor here in biosensors and bioelectronics, so the same center. And then he went to Davis, where he's had this spectacularly successful career, primarily doing a lot of research on properties of molecules, electrical, mechanical, and others that you can measure and then use in devices that can tell us interesting things about people and our environment around us. And I think he's going to talk about things probably with Raman and other technologies. And so I won't say any more, but let's welcome him and I look forward to hearing his talk.
All right, thanks. Thanks very much. Thanks for the introduction. It's always great to be back at ASU. Always love it here. It's been great to catch up with some old friends and colleagues and meet a lot of new people. It's amazing how much ASU has changed.
Josh, could you keep the microphone up just a bit?
A little louder.
Thank you.
Is that better?
Yeah, better.
Okay.
Thank you.
Yeah, so it's definitely changed a lot around here over the last 10 years. So it's great to catch up. As Josh pointed out, today I'll be talking about the work that my group has been doing on trying to understand the electronic properties of biomolecules and how we're trying to take that knowledge and actually use it for specific applications. And so the first question you might ask is just, why would you want to study the electronic properties of biomolecules? And I will say there are a lot of potential answers to that. I will start by just giving a couple. I'll start with the most conventional answer to that. And that really stems from the semiconductor industry, right? So this is Moore's Law. And if you look back over the last 30 years, transistors have gotten smaller and smaller. And if you project it forward another 10, you get down to this sort of size scale of about 1 nanometer. But if you look at what's really happened, you get a trend that's more like this. And so we've hit about 12 nanometers and we've saturated there. And really nobody's expecting to go much below 12 nanometers. And so you may say, well, that's really pretty good. That's about, on the order of 100 atoms in the cross section of the gate length. But if you think about the full 3D volume of these kinds of devices, you're still talking millions of atoms, right? If you compare that to molecules and biomolecular systems, they naturally reside down here at this sort of nanometer size scale. And so you may have a complete device that only has 100 atoms in it, right? So if you like Feynman, you might say, well, there's still plenty of room at the bottom, right? We still got orders of magnitude we could go there. And so that's been really a push in this area for quite a while to say, can we build electronics out of molecular systems? And part of the answer to that is yes, but I actually think that might not be the best question to ask. I think a much better question to ask is, are there regimes or domains where molecules and electronics can complement each other for specific applications that could be in any number of domains, right? That could be in computation, electronics, and memories, but it could also be in diagnostics and sensing. And so I think there are places where electronics can complement biomolecular systems as well as the other way around. And so if I just give one example of that from a classic molecule, right? Everybody's favorite, well, if not anymore, DNA. I think we're all comfortable with the fact that DNA is a memory, right? We each have about 3 billion base pairs. There's nothing particularly special about that. Mice also have 3 billion base pairs. This fern that's been around for millions of years has a genome that's 2 orders of magnitude larger than ours. And so people said, well, instead of genetic information, can we store digital information inside the DNA? And so you just assign some bit values to each one of the bases. This is an approach that was really popularized by George Church's group. And what he showed is that you could take something like his book, you can digitally encode it in DNA, and then basically in an afternoon, you can do some PCR and you can make more copies of this book than any book that's ever been published, right? So he was able to overtake the Bible in like a few hours. And so certainly you can store digital information in DNA, but the question is there advantages to being able to do that? And well, there are certainly some advantages. Here, you're storing information in covalent bonds instead of in electronic charge states. So it can be extraordinarily long lifetime, right? People have found DNA just this year in caves and been able to sequence Neanderthal DNA, and people have shown that with like mammoth DNA. And so you can have, in pretty sub-ideal conditions, very long lifetime storage of these types of materials. The data density for DNA is far superior to any of the solid state type of solutions that we have today. Depending on how you count here, it's at least 6 orders of magnitude better. And another advantage that I think is a little bit less obvious is just how energy efficient this process is. So today, each one of us will produce about 100 billion new nucleated cells. Each one of those cells has about 3 billion bases in it. So if you run the numbers, each one of us is going to copy an exabyte of data today. And we each do that while running at 100 watts, and we use a small percentage of our total power for doing this, right? And what that means is that just the people in this room will copy more data today than all the electronic systems in the world will produce, and we'll do it by using less energy than this projector that we're looking at, right? So it can be really extremely energy efficient. The problem, of course, from a technology standpoint is it electrically readable? The way DNA memories work today is you store that information chemically, doing chemical synthesis, and then you have to do sequencing to read it out. Sequencing has improved a lot over the last couple of decades, but if you compare it to the read-write times on my laptop, it's still painfully slow, right? So if you want to get to the point where you can use molecules in electronics or be able to read out electrical information from molecules, you need to be able to find ways to connect those two together and sort of solve this problem. So this is sort of the underlying question of the kinds of work that my group has been doing over the last several years. And so to answer this question, can you electrically read out information from molecules? Really, there's 3 sort of underlying issues or problems you need to think about. The first one is just developing a fundamental understanding of the way molecules behave when you make contact to them with electronic materials, right? So if you take them out of their sort of normal environment and put them on a surface and start applying fields and currents to these things, things are going to change about the way they behave. And you need to understand what happens there to be able to get back to the original biological information, right? If that's what you're trying to do. The second thing is trying to find areas where you have specific technical advantages of combining electronics with biomolecules, right? Are there places where you have direct advantages over what's conventionally done to be able to push towards these low-hanging fruit, right? These initial applications that allow us to speed up the movement of the field. And the third area is in systems integration. So here, right, if you can make, here what you want to be able to do is create systems that are robust and reliable enough that you can use them in the field if you want to move towards applications. So can you make contact with these molecules and do it in a reliable enough way that you could actually measure these out in the wild? And so my group really works across this entire scale. And we do that because that also provides feedback in the system, right? As you develop new integration tools, that allows you to develop new integration capabilities, which also informs you about what applications are available. And then doing both of these things tell you what you really need to do to be able to do systems integration. And so that allows you to continuously improve on all 3 domains. And so with that, really my goal today is to show you how these 3 thrusts all work together by giving you some examples of the kinds of work we do in each one of these domains. So I'll start by talking about the work we're doing really at this very fundamental level. We've been working on developing a new system that allows us to look at both the electrical properties and do Raman spectroscopy on a single molecule simultaneously. And so I'll talk about how we do that and what kind of information we can get out of that. At the application stage, I'll talk about some work we've been doing on pathogen identification using RNA. And finally, at the systems integration scale, I'll talk about the work we've been doing on trying to leverage a lot of the impressive progress that's been taking place in DNA nanotechnology and DNA origami structures, to be able to create systems where you can interface these biomolecular systems with electronics in a more reliable way. So with that, I'll start here by talking about the single molecule Raman system. So conceptually, this is sort of the goal, right? We want to be able to put one molecule in between two electrodes, be able to do sort of conventional electrical measurements on these things, measure the current, measure the conductance, and simultaneously get a Raman spectrum from the system. Now, we chose Raman for quite a few reasons. One is it gives you a chemical fingerprint of the molecule that you're looking at. So it really allows you to verify that you know exactly what you're looking at. And things like TERS, tip-enhanced Raman spectroscopy, have been around for a while. So we know that it's possible to get down to single molecule level resolution with these kinds of systems. And so if you can do that, there's a lot of potential applications from that, right? Raman has become one of the premier spectroscopy tools for 2D electronics materials. So it's been accepted already by the electronics community. There's a lot of physical things you can learn about it, molecule-molecule interactions. It actually is an inherent probe of the local temperature of systems. And it's also used widely in the pharmaceutical community and in the biological communities. So it's a system that is being used across a lot of fields, which will also help with adoption of this kind of system long term. So that's why we want to do Raman. I guess the next question would be, how do you actually make contact to a single molecule? And so for this, we use an approach that was actually developed by N.J. Tao almost 20 years ago. This is a single molecule break junction system. And the way that it works is you have two electrodes, you apply a voltage between them, you measure the current, and then one of the electrodes, you have to have sub-angstrom level mechanical control over it. So you have to be able to move it very precisely. And then all you do is take them, drive them into each other, pull them away while you have the molecules you're interested in the vicinity. And then sometimes you'll bind one of those molecules. So you get traces that look like this. Sometimes you don't bind any molecules. And then when you do, you get these sort of step-like features. You can do this very rapidly, so you can get thousands of traces, and then you do statistics on it. So then you can get a histogram that looks like this, and then you can say, this is the most probable conductance value for a single molecule. Once you have that, you can go back to your original data and have pretty high confidence that you're looking at a single molecule here. Once you know all this, then you can take it a step forward. You can add feedback to the system so that you recognize those plateau features in real time, and then you can stop and hold the molecule. And then you can do things like sweep the bias, apply a gate field, do mechanical perturbations, or, as I'm talking about today, do Raman spectroscopy. So conventionally, so when NJ first invented this, he used a scanning tunneling microscope for doing this. And that's really the most common way still to do those kind of measurements. People have developed a few other ways, but they're all sort of laboratory grade equipment. If you want to move to the place where you can do these in a field or in a chemical laboratory, really what you need is something that's going to be much more robust and in a much smaller form factor. And so something that we've been working on now for a while is to develop a chip-based form of this type of measurement system. So this is a microelectromechanical system. So it's a single chip system that's designed for doing these kind of break junction measurements. And the way it works is you have these banks of electrostatic capacitors that are suspended with a spring structure. And then when you apply a voltage between them, it actually drives this shuttle back and forth in this direction. And so then it actually generates enough force to break this little gold structure here. And this is where the two tips form that you have the molecules that you look at. So these two tips, the one tip just moves back and forth to do the tapping. So this is what the system actually looks like once we've fabricated it. So you actually have two sets of these capacitors. We have some sensors there so we can run a closed loop. And then this part right here is actually the bow tie structure that we break down where we do the measurements. So this has a lot of advantages over the previous system that we were working with. It's small and that makes it actually a lot more stable, but it also makes it so that it's easy to integrate with other types of characterization systems. So here we can take it and actually put it right under the objective for our Raman microscope. So you can have a high objective lens, you can collect a lot of photons, and so you can actually capture Raman spectra with pretty high temporal resolution. So now the way these measurements work is we take this tip and we separate it by a small distance. So we're in a tunneling range, and then we just wait for the molecules to diffuse into the gap. And when that happens, we'll see these sort of blinking events. And so this is the conductance for that molecule. And what you'll notice in the Raman up here is when that blink occurs, there's this huge change in the intensity of the Raman spectra, right? So you're actually getting spectra from that molecule in the junction. And if you look at the individual spectra, you can actually see the huge change in the spectra over the background when you have these binding events. And when it breaks down, you go back to basically zero in the field. So that's how we do the measurements, just to give you a little bit better sense of what's happening. This is a movie showing an example of what happens. What you can see is there's a lot of changes in the spectrum over time. And so what these changes, so I guess an important question there is, are these changes that you're seeing in the spectra just noise, or are they actually telling you changes in the molecular configuration as you are holding the molecule? And so to answer that question, we collected tens of thousands of these types of Raman spectra. This is an example of about 10,000 of these spectra. And then we use machine learning to try and cluster them into cases where we know the molecule is present and when we don't. And once we have that, we can actually look at the spectra and identify specific features that will tell us when we're looking at a single molecule bound between the two electrodes. And so you see there's lots of changes in the spectra between the cases where we have a molecule bound and when we don't. But the most prominent one is this one here around 1550. And this is actually a vibrational mode of the phenyl ring in the molecule. And once you have this, right, you can do some theoretical modeling and see that once you apply gold atoms to the sulfurs on this biphenyl dithiol, sure enough, you get this change in the intensity and the shift in this ring vibration mode. So it really does tell us that the presence of gold atoms on both ends is a sort of direct indicator of, this is a direct indicator of when we have a single molecule bound between the two electrodes. So once we have identified a mode like that, then we can really start to get into details of what these configurational changes mean in the system. So this is one example of the kinds of things you can do. This is the full spectra during this blinking event. And this is that ring vibrational mode that I was just talking about. If you zoom in on it, you can see there's this sort of systematic change in it. And what I plotted here on the bottom is just the change in the peak position. And you'll notice how well correlated these changes are with changes in the conductance, right? So here there's this discontinuity, this change in the conductance. And similarly, there's a discontinuity in the Raman vibrational energy. And so what that allows us to do is extract a conductance value for each Raman value. And what that's really telling you is what the strain inside the molecule is as a function of time. Once you get to this level of detail, you can actually simulate the molecule systematically as you separate the two electrodes. And then you can start to figure out real sort of subatomic level details about what's going on inside the molecule. So if you look at this changes in the peak position, what this is really telling you is at the beginning, you have a specific separation between the two gold atoms that are bound to the molecule, and you will only get that vibrational mode if you have the strain that's related to this separation. Then the electrodes sort of drift apart over time. And then when this discontinuity occurs, what's happening is you're getting a change in the gold configuration that causes the gold atoms to relax, but then causes a compression of the molecule. And then the electrodes continue to separate away until eventually they break. So you get very sort of low level detail about what's going on in the system. And so this as it is, will be a pretty useful system for us for trying to understand these sort of very fundamental details. But it also is going to serve as sort of a launch pad for moving more towards biomolecular systems and opening up some new applications. And so just very briefly, I'll give a couple of examples of where we're heading with this system now that we have it up and running. So one thing that we're going to look at is complex proteins. So this is azurin. It's a copper complex that's involved in the cytochrome chain. So it's a nitrogen fixing bacteria. And it's got this simple copper complex in there. So it's an electron transfer protein. And people have been studying this for years using a lot of various techniques, photochemical approaches, solid state systems, et cetera. And they've learned quite a bit about the way this molecule works. But in all these cases, it's actually very difficult to get configurational information out of that's driving the electron transfer processes that they see. And so to do that, really what they've had to go do is depend on theoretical modeling. So people do molecular dynamics simulations. And from those, what they found is there's a couple of different binding domains. So this azurin protein can bind through this hydrophobic end to a gold surface, or there's a couple of cysteines here that could also bind to the gold surface. So there's a couple of different configurations. So now that we have this Raman tool, can we actually start to tell what is happening that gives us the electron transfer processes that we measure? So here is an example trace. So I'll say this is hot off the presses. I included it specifically for Stuart's group because I know they've been working on electron transfer and proteins for quite some time now. And what you'll see is we get a blinking event, and sure enough, we can get a Raman spectrum from that molecule. And if you go through the literature, you can identify specific modes, so you can tell what's going on there. And if you do other blinking events, you will see that you can get conductance values that have a much lower value here, but still get a very sharp Raman signature from them. And if you look at the difference of the Raman spectrum in this case, you'll see some of the modes line up very well, but conspicuously, there's a shift in the copper, and there's also the disappearance of this mode here. And it shifts, there's now appearance of a new mode here. And what those actually are is, in this case, this is the energy for a sulfur gold binding, which tells you in one case, you're actually getting a gold thiol bonding to the system. And here, you don't have that, but you do have the gold nitrogen bonding. And so it actually does tell you the different binding domains, right? So if you go back to what I was just showing a minute ago from the theory, this case, you should have the thiol bonding, and that should give you a smaller system and a higher conductance because you have better coupling. And in this case, you have the hydrophobic binding, the distance is longer, and you have a lower conductance just as you expect. So this combined Raman single molecule system will actually start to give us very detailed information about way charge transfer occurs in these biological systems. And of course, there are a lot of other things we can do with it. We can start to look at biomarkers, try and do things like identify different amino acids, different peptides. Just to give one more example here, if you look at cysteine versus glycine versus tyrosine, you can see you get links for all of them. They give you different conductance values, but you also get significant changes in the Raman spectra. And those do match in general with what you would expect from the theory for the Raman spectra. So it gives you multi-dimensional spectroscopic information about what's happening inside these systems. And so this is a system that I think long-term will be able to work on both fundamentals and start to push out to some new application spaces. And so I hope from going through this, that sort of gives you a first instance of understanding how these systems work together, right? We developed this new integration system, the MIM system, that allowed us to do new characterization, and it started to allow us to get into new applications, right? So what I figured I would do next is, instead of these very short snippets of the applications that we see going forward, I'll talk about one that we've spent a few years on. So we've been trying to do pathogen identification for a long time, for the last few years. We started with E. coli. We actually had a startup in that domain. But then when COVID came around, we saw that as an opportunity to try and apply this to a completely new system. And so what we've been trying to do is actually identify COVID variants using single molecule conductance measurements. So COVID, of course, doesn't need any introduction, and I think everybody is very familiar with this curve of the number of traces worldwide with the multiple peaks that we have. Now, Omicron showing up, we may be on the cusp of another wave. And some people said, well, this just means we've done a poor job of flattening the curve, but it actually also shows very interesting correlation with the rise of variants, the emergence of different variants. So two of the very first variants that of interest that showed up were what were the so-called New York variant and the UK variant. And they started to show up just about this time last year. And then by February and March, they really started to dominate the cases that we were seeing worldwide. And right after that, we started this other peak. Then about March, April, the Delta variant started to show up. And by June and July, that totally dominated the landscape. So what we decided to do was try and identify a couple of these variants. Now, these variants occur because there are usually single, sometimes multiple changes in individual amino acids in the spike protein, right, in this binding domain. And so the New York variant is actually called E484K. And what that means is that the 484th residue, this glutamic acid, becomes a lysine, right? And then if you look at the genome, you can actually go through and find the 484th position, and sure enough, there's an E there, and the same thing for the UK variant. And so the question was, how can we actually identify these things electrically? So we've been studying pretty short DNA sequences now for some time, and RNA sequences. And so what we know is that if you have something that's on the order of, say, 12 to 15 base pairs, we can measure the conductance of it relatively easily. And that's also long enough that it's pretty specific, right? So that same sequence shouldn't show up elsewhere. And so what we did is just look at a few bases on either side of where this mutation takes place, and then took the complement of that and made that a probe molecule for ourselves. Right, so what we have here for the UK variant is we took the complement of that sequence that I just showed, and then put a sulfur on either end of it so we can bind it to gold. And then now that is complementary to the RNA genome of the UK variant. And so if we can measure the conductance of this, that tells us that the UK variant is present. Now we need to be able to distinguish that from any possible interference, right? So the wild type is only different by a single base. So the conductance of this one has to be different than the conductance of this one. And it also turns out, if you go through the whole genome, there's one other place in the genome that has a base that is a single mismatch from our probe. Similarly, for the E484K, we went through the same exercise. In this case, there's two possible mismatches, right, on top of the wild type. So let's take a look at what happens. We do these break junction measurements I showed. We basically just put it in solution, do the tapping measurement, look at the conductance. Sure enough, we can measure very clearly a conductance peak. If we take the wild type and put it in there, again, we can get a measurement. And the same with this genome mismatch, right? And so the good news here is there's a clear shift in the peak position. The bad news is that the distribution is quite wide. But if you do enough measurements to be able to get statistical analysis to be able to do a fitting, right? So you can't do one, right? You have to have enough measurements to be able to get a fitting. But if you get a fitting, the peak position is specific enough that you can identify it versus the other cases. So you can actually tell the difference between these three. Similarly, for the other variant, same sort of story here. It happens to be even broader, but the conductance value is actually pretty similar for the perfectly matched case, the target case. The wild type in this case actually causes an increase in the conductance. And the same is true with the genomic mismatches.
So Josh, do you have to take, how much data do you have to take? And specifically, I'm thinking of being competitive with fluorescence type measurements, which of course have to use many molecules. But if you're in that same limit, then you haven't necessarily got an advantage and you have an expensive piece of electronics versus a bit of paper. So
Yeah. So the fluorescence measurements, I guess it depends on what kind you're thinking. If you're talking about trying to do amplification, right, we don't need to do any amplification. So the number of measurements that we need is basically if there are on the order of 1,000 copies, then no problem. So we've shown this for the E. coli that we can actually get down to attomolar concentrations and still get enough measurements that we are able to get a peak in the histogram. Now, what we're doing, I'll show a little bit of, is trying to use machine learning techniques to improve that even more so that you can say, if you have 10 measurements, does it come from this case or this case?
And with attomolar
Yeah.
concentration, how long do you have to wait for the junction to capture something?
So here we're doing capping measurements. And so in the order of a half an hour, we have sufficient amount of data to be able to do the collection.
But if you're attomolar, doesn't it take a long time for something to diffuse to the junction?
So our hypothesis there is that the field is actually playing a helping role. So we don't, yeah, so I don't understand exactly why it happens so quickly.
But that's nice.
Yeah, but it does happen that quickly. So we've been trying to do some modeling. We think it's because there's a field-assisted electrophoretic effect there. But I'd say so far that's just a hypothesis. So in this case, the peak position is sufficiently different that you can identify the perfectly matched case, but the three mismatches are pretty hard to distinguish, right? But really all you have to be able to do is say, do I measure a peak? If so, you have COVID. Then the second question is, can you distinguish between these two? And like I said, these are genomic mismatches. So what you really have to be able to do is differentiate the case between where, say, 3 of these are present and when 2 of these are present, because these 2 will always be here, right? And so what we did is just went with the worst case scenario. We said, okay, we may have the UK variant, the wild type, and its mismatch all there at the same time. And if we do that kind of measurement, we get this very broad distribution in the conductance histogram, right? Because the individual peaks are so broad, you can no longer distinguish between them. But what we want, and the same thing is true for the other case, right? But what we want to be able to do is distinguish between the case where we have 4 possible targets or 3 possible targets, and we don't have the target of interest present. And what you can see is there's clear shifts in the histogram when those happen. And so this is where the machine learning that I was talking about is coming into play. So if we train the data on the New York variant and then try and do these measurements, we have about 80% probability of being able to identify between when the UK variant is present and when it's the wild type. And in the other case, because the conductance is more significantly different, we can actually get close to 100%. So you can really identify the difference in those cases. So that's just one example of the kinds of things that we're trying to do to move towards applications for these single molecule electronic measurements. The final area that I wanted to talk about is how do we do systems integration. So we've been working on a few different approaches there. And the one that I'll talk about today is the stuff we've been doing with DNA origami. So probably for this crowd, I don't need to spend much time describing DNA origami. I think everybody's probably familiar with it, right? You take this long scaffold strand, you have a short staple, and then you can start, if you have enough of those staples, you can basically fold it up into practically any shape that you want, right? With extraordinarily high resolution, right? Coming from an electrical engineering background where we struggle to make things that are tens of nanometers, here you have 2 nanometer scale sort of precisions. And of course, there are a wide variety of different approaches here. People have made DNA bricks and DNA tiles. So the question we wanted to ask is, can we use these to either control their native electric properties, use them to template other electronic materials, but most importantly, put them where we want them on surfaces to be able to actually interface with electronics. So we started by just trying to understand some of the native electronic properties of these systems. So we started with the smallest origami we could. This is just 4 helices all tied together. We have a couple of file groups on either end. And so it forms this sort of square system that's about 10 nanometers long. And then we do the break junction measurements on it and measure the conductance. And what we get here is a conductance that's 10 to the minus 3 times G naught. So if it's a perfect 1D channel, the value we would get is 1 G naught. So it's not a perfect conductor, right? But just to give you some sense of what's going on here, if you remember the conductance values that I talked about a minute ago, they were more than an order of magnitude lower. So if you look at just duplex double-stranded DNA, this one's only, 11 base pairs, 12 base pairs, is more than an order of magnitude worse. So it seems that these are pretty highly conductive, which was a bit of a surprise over these sort of length scales.
Josh, do you have any idea where the very high background comes from?
So the red here is what we have if we're just in buffer,
Right,
and then the black, in general, we have this sort of number of counts whenever we're looking at things in aqueous solution.
Do you know what they are? You've obviously got some specific connection in the middle there where you've drawn your Gaussian.
Yeah.
You've got those wings that just go on forever.
Yeah, they seem to even go on forever. And then as one of my last papers with NJ, there's actually a water peak also up here. And so there can be other sort of non-specific bindings that show up. In this case, I don't know if it's just tunneling through the background, right? So when you have all these DNAs on the surface, even when you're withdrawing, you get these longer exponential decays that are present. And so we could be picking up some of that. And then there are also, right, really, the conductance can be anything. And so you're really looking at a large distribution overall, right?
Yeah, I'm just surprised that if this is something attributable to this specific structure, that you get all these other modes of conductance that are spread out so evenly.
Yeah, but I think we see those in the proteins and in other DNAs as well, right? There's always a pretty large background. Even in this DNA, right, you're seeing this sort of background level. And where exactly that comes from, I think is still largely an open question, but I think part of it is because sometimes you're tunneling through just solvent and sometimes you're tunneling through parts of DNA. And so there's this,
We should talk about this
Yeah.
later, but with the use of specific contacts, we don't see backgrounds.
Which yeah, we can talk about.
Yeah.
But I'm very curious then about what
Yeah.
kind of contacts you use without wings, because one of the first, right? So this was surprisingly high. So my student brought it to me, and I actually said, "No way, right? Something's got to be wrong. Go do your control experiments." And so the first thing he did was just try. Oh, there we go. To do things where we don't deprotect the files, right? So we leave them in an oxidized form so they can't bind to the gold. When you do that, you get some background, but you don't see that peak position anymore. And if you put a file on just one end or the other end, then again, you don't see this conductance value. So it's something that requires that we have contacts on both ends. There has to be some sort of mechanical contact to be able to do these measurements. But, right, this is a big structure. And so there's also this question of, can we be binding across it in some other way where it's laying flat on the surface and we're actually measuring current through in another dimension. And so if you look at the individual sort of traces, these are all actually pretty short compared to the length of the molecule. This is a 10 nanometer long molecule. And if you look at the statistics of this step length, by the time you're at 2 nanometers, almost all the junctions are broken, right? So how can you prove that you're really measuring something over 10 nanometers long? And so what we did here is use a modified version of the technique I showed earlier where we had these sort of blinks, right? So we just park the tips at a pretty large distance between each other. And then we stochastically wait for some binding event to occur. And then once we have that binding event, we very slowly pull up the tip to see when it will finally break. And so in these cases, you can get traces like this that go out to 10, 12 nanometers. And you can get enough of those to do statistically. You can see there's this sort of downward trend where initially the conductance is high and it sort of decays over a nanometer, but you always have this sharp breakdown at the end, which really means you are measuring through these sort of long structures. And we have to have the files present to be able to do those. So it means we're not measuring through some sort of random structure that's just forming on the surface. And so this gives us quite a bit of hope for the way origami might behave as a conductor, because this is pretty high conductance for 10 nanometers. And so we decided to try and push even farther. And so we went to these very, very long DNA origami wires. So these are 1.25 microns. They're made by Yonggang K's lab at Emory University, who's also a Biodesign alum, who's from Hao-Yan's group. And so these have this 10 helix bundle sort of structure, so this sort of double barrel structure. And we created a new chip for being able to do these kind of measurements. They all have fluorophores on it. So you can actually identify when individual or multiple origamis bind across your two sets of electrodes. And then if you're very careful with the surface chemistry, you can actually start to measure the conductance of these. So if you just leave this on something like nitride or oxide, you basically get nothing in the background. But if you change the surface treatment so that the surface interactions don't just flatten out the origamis, you can actually start to get conductance measurements. But these are many, many orders of magnitude lower than what we're getting in the short cases. So there's still a mystery there of what the changes are when we're putting these longer systems on the surface. But as I said before, our goal isn't just to measure the conductance of the origamis. What we really want to do is try and place these at specific locations, not just drop them on electrodes and see how they behave. See if we can push these to be at specific spots on surfaces so that we can use them for integration long term. And so what we decided to do here is use dielectrophoretic trapping to try and put these between specific sets of electrodes on the surface. So what happens here is you have a very small gap. So we have 400 nanometers, 600 nanometers spacing between these electrodes. You apply a high frequency AC field on the order of megahertz. And if you have a polarizable molecule in solution, it will actually create a force that's transverse to the polarization axis. So it'll actually force this long rod to actually land in between your two electrodes. And so, well, I won't talk about the modeling, right? But you spend some time trying to figure out what kind of shape and structure you really want to be able to do that. This is the chip that we developed for doing that. And because the resistance is high, we focused on using a capacitance measurement to actually try and detect when we're landing these things between the electrodes. And so the way the measurements work is kind of like this. You apply the high frequency field for about 5-6 minutes. And then when you first drop in the solution, we start that as our capacitance at 1. And then the capacitance creeps up over time because there's a lot of ions in the solution. And you'll see there's some noise and some plateau features. And then once you've done this, then you can go to the fluorescence images and you see a bit of a menagerie of different things there, right? So you'll see some cases where you think maybe this is one, some cases where you see a lot of stuff, some cases where maybe there's a couple, some cases where there's clearly some aggregation present. Then if you go to the STM or the AFN, you can actually go through and find cases where you have these sort of individual wires in between them. The question then is, can we correlate changes in our capacitance measurements with the fluorescence measurements to be able to really identify these kind of cases directly? And so this is something we've been trying to work through. And what we found is that if over this course of 5 minutes, you have a very small capacitance change on the order of 50% or less, then you have just a single or sometimes a few origamis present. If you have a capacitance change on the order of 100% during that time change, you've actually captured many origami wires. And then if you get huge capacitance changes, then you have this aggregation effects. And so it's starting to give us a vector to being able to push down to capturing just small numbers of origami wires between the electrodes, right? So this will still require optimization to get to the point where we're really reliably just landing one at a time. But I think as we continue to push forward on understanding what the conductance is, that will help us to really refine this sort of measurement. So with that, I've covered a lot of ground today, and hopefully that gives you an idea of how these different areas fit together to push towards doing applications for biomolecular electronics, right? I started by talking about all the Raman stuff that we've been working on and how we can use this for looking at things like proteins and amino acids. I showed one of our initial application spaces here trying to do pathogen detection and talked about the stuff we've been doing with COVID. And finally, I've talked about how we're doing integration with origami systems, right? And so our goal is really to continue through this kind of process to push these towards applications. With that, of course, I want to thank my group at Davis, right? Mashari and Zara have been the ones working on the COVID detection. Luke and Hawk were the ones doing the Raman and the MIM system, and Tahir and John have been focused on doing all the origami stuff. And of course, it's very interdisciplinary work. So we have a lot of collaborators that work on these kinds of things with us. Of course, we wouldn't get any of it done without the funding agency. So thank you very much for your time. I'm happy to answer any questions.
All right. I'm gonna ask them real quick. So with the origami wire, it was configuring sort of as diagonal across the.
Yeah.
And it looked like that was always happening. Is there a reason for that?
So, yeah, they're not always that way. I should probably generalize the pictures that I'm showing you a little bit, but it does happen a lot. Now, I think what's happening is just depending on where it's coming in the field, it like hits one side and sort of eventually sticks down on the other side. So they're never straight. Yeah.
Okay, yeah.
Yeah.
Yeah, for this origami system, I wonder if you have thought applications for studying interactions with drugs, DNA.
Could you repeat the question?
Yeah, we're recording and I forgot. I should use the microphone too.
Yeah, so the question was, what applications have I thought about on the origami stuff in terms of like binding DNAs or doing drug discovery? I'll be honest, I haven't thought much about on the drug side. Definitely, I think if you can have those, right? So sort of the goal there has been to use those to put things down like graphene nanoribbons at specific spots so that you have a conductive pathway. And then once you have that, then I think it really opens the door to being able to do sensing pretty easily, right? So if you have a DNA and you capture something, you're going to get a change in the charge transport properties, right? So if we can improve the conductance by a couple of orders of magnitude more for the origami structures themselves, which I think is doable, then even there, without going to something fancier like graphene nanoribbons, we should still be able to measure these sort of changes when there's a binding event.
We're going to bring the mic over to you.
I've got a loud voice, but anyway, Josh, first of all, from someone in the field, beautiful, lovely,
Thanks.
lovely range of work. In your origami DNA wires, did you have chemical attachments at the remote ends?
Yes, yeah. So we do have thiles on those ones. So we've tried it both with and without thiles, especially with if we do those on nitride surfaces, it's actually not too much of a problem. There's enough non-specific binding that they stay. But if we do that on the sapphire ones that we're doing the dielectrophoresis, basically they don't stick well enough. And so if you don't have the thiles, you lose them all by the time you try and go do fluorescence.
And I just wondered, have you done controlled experiments without thiles to see that you presumably don't get any current?
Oh, you're talking about the, oh, yes.
Origami, yeah.
For the long origamis. Yeah, if we don't have the thiles, we don't measure, we don't measure the current in those. Yeah.
Yeah, very nice talk. I have a question about the sequencing, because you showed us the short pieces of DNA. So how you would practically do that? Let's say now we have this Omicron variant and everybody wants to know as fast as possible how fast it is spread.
Yeah.
So would you be able to identify from a patient sample if he has Delta or Omicron?
Yeah, so I think we should be able to, right? So we definitely still have a lot of development work to do. But the idea is the approach is agnostic, right? So if you have a specific sequence in known interference, you should be able to identify between any of those, right? So once you know what the conductance value is and the conductance value of the interference, then you can tell the difference between any of them, right? So the part that we've been working on, and that was a big part of what had been going on in the startup, was trying to do sort of the pre-processing part, right? The biochemistry part. Because you have to do sort of like a mini prep to get the DNA out, to get the RNA out, and then you have to be able to bind the probe to the right sequence of RNA. And then the issue that we were having is that sometimes the long strands of RNA interfere with being able to make contact, right? So after that, we've been trying to find the right nucleases to use to digest down to just that part. And so then once you do that, the measurements themselves are actually pretty quick.
So the preparation takes a while.
So I think, it doesn't necessarily have to. People have worked out a lot of those things on systems on a chip. The part that we haven't worked out is finding the right nuclease, right? Which, as you know, can be a challenge, right?
And then I have actually a question about this quantum biosystems.
Mm-hmm.
So it sounds always so cool to call something quantum, but when you talk with a quantum physicist, he would have problems to really see the connections. So how you could make the connections between quantum and the cool systems you have shown us.
Well, I will say, I did not name that company. That company was a collaborator and a funder of my group.
Okay.
It's
Right.
dead now, isn't it?
Yeah, they just EOL'd, yeah, so they weren't quantum enough to survive. But yeah, they were actually focused on doing more like sequencing of DNA.
So probably you already answered my question, but just in case I didn't understand correctly. So for the short RNA detection in your experiments, did you use short RNA or RNA from COVID?
So all the stuff that I showed today, well, especially with the COVID, everything I showed was short RNA. Synthetic RNA. Right? So for the E. coli, we made it farther to actually go through some digestions and things, but we're not BSL-2, right?
I understood. Mine neither. Okay. But have you tried extending the synthetic RNAs that you detect and that change the conductivity or no?
So if we did that, if the RNAs that we're binding to are less than 40, 50 bases, then it didn't seem to bother us too much. If they're much longer than that, then the yield went down quite a bit. We could still do it sometimes, but it wasn't reliable anymore.
Thank you.
Yeah, so thank you for your talk today. So I was recently hired to work at ASU. I currently make conductive hydrogels for various biomedical engineering applications. And I've been currently using conductive AFM to measure conductivity of these hydrogels. But I'm curious about the single molecule Raman technique that you mentioned, as well as the chips that you've been fabricating. Since my hydrogels are typically a dispersion of conductive additives within a biomaterial matrix, do you think that it could be a candidate for that type of measurement or method for measuring conductivity, the Raman and your chip method, which is, I guess, from what I saw, is typically a covalent network for measuring conductivity versus a dispersion in a hydrogel system?
So I think there are definitely things that we could do in that domain, right? So I don't know about doing the break junction type of system on the hydrogel, but some of the nano gaps that we had, putting the hydrogel on it, we could certainly do electrical measurements and Raman measurements at the same time. And then there's some interesting things you can do about trying to map out charge transport pathways and things like that in the system. So I think there are places where that combined system could help for something like that.
Thank you.
Very beautiful work about the Raman part. But did you test the Stark effect on those devices? Like if you switch the bias, polarity, or increase the field, do you see the shift of the peaks at all?
Well, so two parts to that. If we do a bias sweep, then we do see some small changes in the modes. We haven't done it systematically enough to see if it's changes because of the bias or just because of the drift of the electrodes, right? Because these are all at room temperature, so there's always some drift there. But what we do for sure see is we can also look at the anti-Stokes, and you definitely see pumping of the anti-Stokes that changes with bias.
Thank you.
Yeah.
My question is simple. It's just by moving your break junction system from AFM to wafer, how much relaxation you got, for the environmental requirement, the noise from surroundings? Is it much easier? Or?
It's not much easier, right? So the question was how much better is the vibration isolation on the chip versus doing it in an STM or type of system? It's better. But I think it's still been a little bit, I don't have a quantifiable answer there yet, because part of the problem is, right, just thermal drift, right? And so how do you tell exactly how much of the instability you're getting is due to thermal drift of the electrodes versus how much of it is due to measuring room vibrational noises that are excited, right? And so it's definitely better. You can capture stable junction easier and leave it for a few seconds, but then also the gold bonds break, right? Or the gold bonds break. And so there's sort of this limitation of how well you can measure the stability.
the whole thing become a single piece, right?
Yeah, and so you don't get the differential movement
Yeah,
between the
right.
tip and the surface like you do in the STM. So that's definitely a help.
So just to clarify, you're looking for an RNase that selectively cuts the RNA, or are you looking for the DNAs?
RNase.
Because, it strikes me that you could target the RNA any way you wanted to by coming in with a complementary DNA and then use RNase H. And then you could
Oh.
cut the RNA any way you wanted at whatever positions you want.
Well, let's talk.
Yeah,
I think that'd be a great
You might be able to
idea.
solve it that way.
Yeah.
All right, so we had a lot of good questions here. This was obviously, you really stimulated the group here. That's
Great.
terrific. Well, if no more questions, let's thank Josh Hihath. Fantastic.